Hi everybody, I'm Dr. Kifari. Today I start biology lecture and the first lecture about the formation of organic monomers. Okay, I start with explanation of the Miller experiment and we would like to answer this question: how was it proven that the life is originated from the inorganic material? Okay, the Stanley Miller is did an experiment in the closed system. He started boiling the water in the closed system, and the water with evaporation, okay, inside this cylinder goes this area, and with two strong electrodes with high voltage, he makes spike. And he put in the closed system ammonia, hydrogen, and water, and methane. And then he used the condenser with the cold water, which is rotate from here. He tried to make a cold this vapor, and condensation happened, and the formation of water, and it came back again in this dilemma. As you see here, this system is like nature. For example, it is like ocean or sea, and it is evaporation because of sun and because of the raining and tender, okay, and in the raining time the water came back, okay, with a high spark in the tender, okay. After repetition of that many times, he collects some sample, he found in the sample organic monomers like some amino acids, maybe some sugar and nucleotide base. But the ribose and fatty acid and the line carbon chain we cannot find in this system. Then this experiment shows in the billion years ago, when in the system there was methane, ammonia, and hydrogen and water, and half of the earth was the sea, and in this case maybe no oxygen, or the very low amount of oxygen. Start the formation of the monomer organic compound, and after that start formation of the big organic polymers, and maybe start life. Okay, maybe first virus can be formed, and then could be formed, and then after that the bacteria. But other important things, mainly this process happen in the sea because of in the sun there is a UV light. Because the UV light is one of the important things, can destroy the life. Then it is important. One of the things is very important, the formation of this process and cells inside the sea, ocean, in the sea. And the other question we can answer this: how the oxygen concentration is changed during the period of evolution. As I mentioned earlier, atmosphere probably contained water, carbon monoxide, carbon dioxide, nitrogen, methane, but little or no oxygen. Then in the system, after several years, because of the process of the Miller, and start the formation of virus, then start formation of the bacteria. There is a three hypotheses shows how the oxygen start production. The first group was the chemosynthetic bacteria. Okay, this compound 4 billion years ago use energy, okay, as a oxidation of the hydrogen sulfate, and in this process the first bacteria which could produce oxygen, okay, it's very important. And the second group, after 200 years, million years later, in the 3.8 billion years ago, anaerobe photosynthetic bacteria, okay, it also helps for production of oxygen. And high energy, only ATP, not the photolysis, and hydrogen of the organic compound like hydrogen sulfide used form of organic matter. And the third group is the cyanobacteria, water hydrogen source from the water, no oxygen accumulation during 2 billion years, very low amount, only in the 1.5 billion years start formation of oxygen, mainly can bind for the inorganic compound like iron too. It can help to formation of oxygen. Okay, then amount of oxygen start, and oxygen in this case when increasing the atmosphere oxygen because of effect of UV, okay, production of ozone can be happen. You see, in this case because of the ozone UV formation, and these systems make a ozone layer which is very important for the protection of the life in the earth. But what is the first importance in question about formation of the life, and which basic chemical compounds, organic compounds we can find in the cells in the animals or plants or in any organism? And one of the important is the triglyceride. Triglycerides are formed by the combination of the glycerol and sulfathy acid. Okay, as you see here, it is a glycerol, or the other, we can say 1, 2, 3 propane triol. It is a triol alcohol. It reacts with 3 fatty acid and form with removal of the water, dehydration, and for remove of this water, and 3 ester bond will be formed, okay? And then glycerol bind to the 3 fatty acid. But we can have a 2 type of the glycerol. One is the saturated fatty acid, 3 type of fatty acids. One is the saturated fatty acid, one is the unsaturated fatty acid. Saturated fatty acid like palmitic acid, it has just a single bond and all of the bonds are sigma, and also it is a sp3, these carbons, angle is 109.5, and also mainly have a higher melting point. The other groups is unsaturated fatty acid, which has a double bond. Okay, it has a pi bond, which is a sp2. The angle is 120 degrees. And as I mentioned, sp3 is the tetrahedral and the sp2 is trigonal planar. But we can classify unsaturated fatty acid for 2 groups: mono unsaturated fatty acid and the poly unsaturated fatty acid. In the mono, we have just 1 double bond. Okay, it can be cis or trans. Okay, as I show you here. Okay, I explain the system in the next page. As you see here, if we have a fatty acid, and we have a double bond here and here, it is a polyunsaturated fatty acid, and both of these fatty acids, because of the hydrogens in one side and the carbon in other side, both of them are cis. Okay, in the mainly cis fatty acid is good for the organism and the trans is bad. But how we can make a trans from the cis? We change position of hydrogens with carbon. For example, the first one, I made this Okay, double bonds here, and again I continue like this, and the other double bond. Okay, as you see here, because of the one hydrogen is up, other hydrogen is down, and the carbon with hydrogen is opposite direction, both of them will be trans. And if we have 6 fatty acid, and if we increase the heat, for example, if we're boiling of oil a lot, we can make trans fatty acid. Then it is a boiling of the oil is not very good and can cause trans fatty acid, which is harmful. But for the polyunsaturated, more than one double bond is important. Okay, for example, if I have a double bond here, this fatty acid has a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22 fatty carbon. Then I can write 22 carbon, 2 point, the number of double bond, it is here 3. And I can show by the delta the position of the double bond, 1, 2, 3, 4, 5, 6, 7, 8, okay, 9, 10, 11, 12, 13, 14, 15, 16, okay, it shows 3 double bond, carbon number, position of double bond. And other things we can show here, we can show by the omega. Omega, the position of carbon from the end, 1, 2, 3, 4, 5, 6, the first double bond is the 6, then we can write omega 6. It is a official name for the fatty acid, polyunsaturated. We can show in 2 ways, and we can show with the omega, and also we can show with the position of number, double bond position, and number of carbon. As I mentioned here, and very important, okay, polyunsaturated can also divide in 2 groups. One is the essential fatty acid, and the other is a non-essential. Essential, for example, type of omega-3, omega-6, and non-essential, body, we don't need, body can produce it, okay? As you see here, I made one fatty acid, triglyceride, from 1 glycerol, plus 3 fatty acid. We made esterification and we removed 3 water. As you see in the structure, glycerol can bind with the 3 ester bond for the 3 fatty acid and the formation of the 3 acid glycerol or triglyceride. What is the function of triglyceride? They are store energy usually, and not component of the cellular compound. How we store of this storage of this compound? Usually in adipose tissue. Okay, how they do it is very easy, by the transfer by the lipoprotein from the liver, usually transport by the LDL, VLDL, and the chylomicron. Okay, they transfer the triglyceride for the adipose tissue. Usually they separate from the fatty acid glycerol. And the other important things, when we need an energy source, in this case triglyceride can break down the fatty acid and glycerol, and the fatty acid can go to the muscle or the liver and can be used for the energy source or the gluconeogenesis. Okay, I can explain a little here. As you see here, for example, if we have adipose tissue, we have a triglyceride. It can go to the blood and the formation of glycerol and the fatty acid. Fatty acid can transfer to the muscle as the convert to the acetyl coenzyme A and formation of ATP in the Krebs cycle, or go to the liver for the gluconeogenesis and the formation of glucose when in the starvation and in the diabetes mellitus situation. And glycerol also can go to the liver, can go again to the gluconeogenesis. Okay, and also we can, when we have a production start in the starvation of diabetes mellitus, mainly this process of triglyceride can go in the direction of the formation of the ketone body. So mainly we can say in the starvation and in the diabetic mellitus. Okay, the next part is what is the structure of the and function of the phospholipid. The most important function of the phospholipid is to form the phospholipid bilayer. In this bilayer, the phospholipids are arranged so they are hydrophilic head, and also with the hydrophobic tails. Okay, and this layer make up the cell membrane and is critical for the cell ability to function. Okay, it has a selectivity in the transport of material from the molecules ions from the cell membrane. Okay, as I say here, we can classify membrane lipid usually in two systems, phospholipid and glycolipid. Phospholipid can be divided in the glycerophospholipid and sphingolipid, and glycolipid can be divided, can be shown with a sphingolipid again. Okay, but different shape. First group is glycerophospholipid. I can show in the structure. It has two fatty acids which can bind to the glycerol, and the last OH can bind the phosphate, and the phosphate bind for the alcohol. Okay, then this two is the head, and glycerol and the two fatty acids which bind together will be tail. Structure of phospholipid I can show here. It has a hydrophilic tail. I write in English, head, hydrophilic head, likes water, and the hydrophobic tail hate water. Okay, as I show you, in the tail we have a glycerol and two fatty acids which bind it, and it has a ester bond, two ester bond, and they are natural, no charge. And as you see the head we have a base that I showed you, alcohol or base. One example is choline. I write all of them. Can be choline, can be serine amino acid, can be inositol, can be ethanolamine or ethanolamine. As you see here, we can show this structure is alcohol base. For example, this structure say phosphatidylcholine. If we change to the serine, it will be phosphatidylserine or phosphatidylinositol and ethanolamine. Base or alcohol always are a positive charge. And the phosphate is a negative charge. Then it will be more polar, because both cation and anion in the head, we can say amphipathic, and it's one of the important things we can show in the structure. As I mentioned, phospholipid bilayer, it is a two-layer, and it is a head, and it is a hydrophilic, and because of this, it has a positive or negative charge. They like water, and water can surround it, and the other side also hydrophilic, and the middle is the hydrophobic, and it's hate water from the glycerol and fatty acids. Okay, and can be fatty acid can be unsaturated and saturated fatty acid. It's very, very important. And I go to the sphingolipid. Sphingolipid has a structure here, and as you see here, and it has a sphingosine which bind to the fatty acid and the phosphate and the alcohol or base, or it's the choline here. Okay, as you see here, very, very important, we have a sphingosine here. Okay, now I want to explain what is the structure of the here and what part we can have. It is very important, and we have to know about this. Okay, as you see here, choline and the phosphate which bind each other, and we have a sphingosine. A sphingosine with one fatty acid, the name is ceramide. Then I can say this area name is the ceramide. And the sphingosine structure, what is the sphingosine structure? I can show here. Okay, it has a carbon chain, CH212, and one double bond, which after double bond we have a OH, and here we have a CH and NH, which bind to the fatty acid. Which bind to the fatty acid. It is a fatty acid. Okay, and this one is a sphingosine. Okay, now these two become ceramide. Okay, ceramide. And as you see here from CH, here we can continue CH2 for glycerol, like glycerol, CH2O phosphate, okay, group, and which bind for the choline. Choline is the ethanolamine with 3NN methyl. Okay, with it is a CH3 positive. Okay, as you see here, it is a choline, and it is a phosphate which bind for the sphingosine. Sphingosine is a carbon chain with double bond and OH group, and which bind to the fatty acid. Very, very easy and very, very important. But sometimes in the sphingolipids, which from the glycolipid family, we have a ceramide. Ceramide again the same. Okay, it is a ceramide, which is the one fatty acid with the sphingolipid bind to the mono or oligosaccharide. It is also we can make with, for example, oligo or monosaccharide. And it is very important you understand how this mechanism can be happen and how the process can be happen, and you have to know how the structure start, and it is one of the important things. Thank you for paying attention. We will continue the next lecture next time. It was the first lecture from my note, and thanks a lot for paying attention, and see you next time.